Yes. Yeah. Okay. So just to put perspective, murburn is a very complex thing. Yes or no? Yes. Nah, but theory is simple, but to nail things, predict things, compute things. Yes. So that's where AI comes in. So today we will have the first instance of AI being applied in murburn. We are going to classify proteins based using AI, and our National Innovation Foundation Director, Dr. Aravind Ranade, he will inaugurate the opening of that database and prediction tool. All right. So that's what is going to happen today. After about half an hour, he will come online, and then Professor Jaeken will give a talk, and then. So, is that ready? 4 minutes. नहीं नहीं इतना काफी है बहुत हो गया एक और चल डाल दे जल्दी so yes what do you think let's Sai, I'll continue your discussion. What are the fundamental principles of homeopathy, say, or what are the practices of, say, acupuncture or Chinese medicine? Do you think they have any basis in the science that as we know? What is the science today? All that? Any discussion on that? 1 second, I'll just, yeah, go ahead. Go ahead. Give them the offer. Yeah.
So you gave one scientist example who was put aside, and he was talking some meaningful conclusions about exploration. In your case, Donald person, some scientist. So he was put aside when he was trying to talk about some sensible science. You gave us one example.
What? Daniel Arnon. Oh, okay. Yes, yes.
So he was speaking some science, and the remaining the community or the leaders, so-called leaders, Western scientists, they
Yes.
put him aside.
Yes.
So in the Western people, they have done that.
Okay.
My understanding is when we Eastern people, without anything, with plain intellectual, or I would say intellectual, then spiritual observations, we gave some theory which is ecologically friendly, and all types of balances have been maintained without changing anything. The same concept applies. They have taken, they have suppressed the concepts of all these fundamental things, where I need not actually I need not really take so much of difficulties actually. Okay, so all these Ayurvedic rishis, munis, all those scientists, they wanted to do something which is right in front of me, right on my temperature zones. For example, my fundamental question about this current scientific research is I have to do in 4 degrees, I have to do in 2 degrees. That is not science. Because if an Ayurvedic doctor does, he takes a medicine from your surroundings, he takes a plant, then you will be inspired to go and search for another plant which may give you a better solution. So that is research that is going to invite and inspire a lot of youngsters. But today I cannot do my research to do such a type of work you are doing, which needs 4 degrees Celsius refrigeration and a particular chemical. Okay, so this is a restriction on science, narrowing down yourself to promote yourself for your dictations or your power role.
I mean,
play.
not me. Yeah, I know.
Not you. I'm talking about why you asked the question. What is the difference between that Ayurvedic principles, and these Ayurvedic principles are not limited to physical manifestations. There is a mental manifestation, there is a spiritual manifestation, and there are different types of intricate linkage between one from the other. At least Yeah. So these three have to be linked, and these three are linked by breath. So if you can start, I don't need to pay anything. The most economical way is control your breath. Every disease whatsoever, how much ever big it is going to be solved. The breath will tell you. The breath is the eye to your disease, and it will tell you what you have to do. Also, it gives you solutions and keeps the surroundings, takes you to those surroundings where that solution is there. So such is the drive, where there is no need of any medicine. I have to depend on some. At the end of the day, human life is to become independent as possible. But these experiments which we are doing in laboratories, laboratory animals, laboratory, all these microbes, all those things, they have a different. We are not at all incorporating one more thing, that is consciousness. Every individual, whether it is dead or alive, there is a consciousness involved in that. So these things are completely kept aside, suppressed, and you want to create a big, a big business empire. You are able to do. So I don't know exactly how to tell this, but we have a very strong platform from which, if we develop further, we can do much more wonders. That's the full theory I have for.
Wonderful, wonderful, nice. Can you hear me? Yeah, good. So, what about Chinese medicine? Now, for example, acupressure, acupuncture. That's something about Ayurveda. What about acupressure, acupuncture?
Those are coming from my.
Uh, yeah, yeah. Suppose now somebody has a pain here, they can press somewhere else, and this can get alleviated. How can the modern practice, that is being modern theories, how can they explain them? Some food, if you eat, our ancient traditions say that that is hot. Some, this is present even in other cultures, even the Aztec cultures. They say that is hot, that is cold. That doesn't have any meaning, any interpretation in our modern context. Then there are later forms of medicine. Have you take my phone? Hmm. Well, no, no, could be. See what I see. Some food. No, some food. See, you see, banana. I was so surprised. So banana, there are different varieties of banana, right? So when I was talking to a native inhabitant, native guy, he said inhabitant. Yes, my work as what was that word? Inhibitor. I am not getting inhabitant. What the hell? Yeah, yeah, inhabitant. So, see, one kind of banana is hot, and one kind of banana is cold. And damn it, it is generally on a statistical scale. It makes some sense because some feel that. So now that doesn't have any bearing with respect to modern context. But what I'm saying is, with all these, see, some people, when I went to IIT Bombay, there was a electrical engineer there who was saying that if you put your feet on the ground, earth, your feet, so many issues can be solved. These kind of things cannot be addressed by a very close-minded view. For me, I like to keep my eyes open, mind open. Certain personal experiences have taught me that there are more things that you can see, count, calculate, and all that. Some things you feel, you experience, and then you know. Until then, some people would always question. But as far as we are concerned, we like to keep things that all of us can generally understand. Suppose if I say this is a microphone, all of us can try to come to a convergence. That's all. So there are different things that modern medicine does not. So, for example, my student Surjit said something about some Ayurveda company being attacked by some Indian doctor, and he was happy. Then I pointed out that that person doesn't know what liver metabolism of drugs, how it happens. He is holding on to some aspects. I don't think that. ancient texts and practices were perfectly right. I neither do I believe that all current practices and theories are. So the point being, nothing was ever perfect, nothing is ever going to be perfect. It's all a journey. So let us try to take information and away. Yeah, yeah. Please go ahead. Go ahead. Yeah.
See, what Vedas and Ayurveda
Please
says,
keep it.
yeah,
Yeah.
small, only
Yeah.
two words.
Small.
The Jataragni is Agni in the Jatar, matlab in the stomach. It is, if it is elevate the Jataragni, then it is hot. And if it is not bringing down the Jataragni, it is cold. It is called a hot and cold concept food.
Yes.
Yeah.
Yeah. So there are many such concepts in the chi, Chinese, the energy. There are so many ideas that evade. Some people say Kundalini, chakras. So who knows what? Yeah.
It is not. Let it first get into the system, then criticize it.
No. I'm not criticizing. What I'm saying is, who is anybody to say it is not? You understand?
Let us say this is it.
No, no. No. Wait, that
Unless I sit down and meditate for
I think I would appreciate that.
can we switch off the AC because I think it's getting cold for you? Yeah. 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 No, we need one more.
and let us
Yeah.
practice it.
Yeah.
And maybe we have to become the rishis. And it takes its own time. That's why I'm talking about adiabatic process.
Sir, yeah. So let us have the quiz now. Sorry to end the discussion so abruptly, but the point that we do not understand, that doesn't mean that it can be completely rubbished. I have seen that when I reported this data, which I wanted to demonstrate right before you, I have already done, but then I don't think you can see it. Even if I do it, it is not something demonstrable like that. I spent the whole day trying to get it done the day before the symposium started. So the some things like dilute things can give effect that people dismiss like that. Some food can be hot and cold. I've seen there can be evidence, but there are problems in sometimes reproducing, but that doesn't mean that it doesn't exist.
Possible to air it.
Yeah, we can. Yeah, we can take a video and it's easily done. So we'll do that. So the point being, all these parallel systems of so-called parallel, yeah, so-called parallel. I said quote. नहीं माइक नहीं दूंगा अभी मेरे को जाना अभी मैं नहीं माइक दूंगा प्लीज अभी नहीं माइक नहीं मिलेगा अभी वी आर रनिंग फार बिहाइंड प्लीज नहीं बिल्कुल नहीं माइक मिलेगा या आई सेड सो कॉल्ड कोट Sir ji, I said so-called cold. Please don't object to that because I did that.
Western people, European
No, let us
people call it
have
alternative.
pride in telling
What
that
is
this is the
alternative?
original.
Original.
And
Yes,
we are the originator.
so-called. I said so-called quote. अरे यार, हद हो गई.
I am adding to it, sir.
So-called.
I am adding to it.
No,
I am adding
as
to
much passionate you are about Murburn concept, I am more passionate about the Indian concept. I want to say this is it.
मैं ये क्या धोती यूं ही पहना हूं क्या? I am also passionate.
Yeah, so let us
Don't worry. So I said so-called. I accept that argument now.
so-called is still a diplomatic way of saying
No, it is not diplomatic. The last thing you can expect me from me is diplomacy. Yeah. Yes. Yeah, नहीं ऐसे कुछ नहीं नहीं एक मिनट लेट्स गो ऑन.
I was just supporting you that parallel alternative. This is a horrible one.
Yes.
Even horrible.
Yes. So let us now we wanted action. Now we'll get some action. Silent kind. Have you put it? It's there? Ready. So put it on. What we are going to do, you tell them. Yeah. Mic, इनको दो mic. नहीं सर जी हमको काम आगे बढ़ाना है ना आपको देंगे ना अभी ठीक नहीं होगा.
Okay, guys. So we have a QR code here. We will have the QR code here, and then you just have to scan it. It will be a quiz. Be as honest as you can. Submit it.
15 minutes,
We'll
na?
give you 15 minutes.
Ah.
Once we display the QR code, and then you start. We already started.
Wait. Yeah.
We already started.
Okay, if you want, you can sit separate. Don't copy each other's answers.
Yeah.
Please. Be nice. No talking. You talk, you're. Yes. If you talk, you're out.
Yeah.
No sharing, no talking,
No sharing, no talking.
no peeping. Be independent. What about QR? Can put the QR, please.
Yeah. So here is the QR.
Anybody does not have a phone? I can give the phone.
3:16 now. We will start the timer. Talk to us. Talk to me if you are having issues, please. Now I am the beautiful person. Sir is not. So.
எப்பா அப்பாவி என் ஃபோன் தந்தா என் ஃபோன்ல பிரைவேட் மேட்டர் எல்லாம் இருக்கு.
Okay. Maintain silence. Please begin your quiz.
No talking.
No talking.
Please.
If you are unable to scan, you can step forward and then scan it. Give me the phone. I will scan that if you have issue.
No talking. It's a very simple task. முத்து கொஞ்சம் இங்க வந்து நில்ல ஏன் ஃபோன் நான் சொல்லிட்டேன் நான் செய்யணும்
ஸ்கேன் பண்ணி கொடுத்துருக்குது
இல்ல கியூஆர் இல்ல அதுக்கப்புறம் நான் இங்க இருந்து போனா நீ என்னோட எல்லாம்
Come on.
இந்த கியூஆர்
Whose was this? Please come back.
You stand here, please. Yes, my phone.
I don't need a certificate, by the way.
Yeah. You don't need. Yeah. Nobody needs any certificate.
And I really like your teaching style. I want to talk to you, but we'll talk to you.
Yeah, sure. That's okay. I know. Yeah. Are you living now already?
No, I'm not able to connect there. I'm gonna.
Okay, go ahead. Yeah, I will. Yeah, sure. I said that because I wanted to hold because the mic. If I keep the thing there like that, the whole thing will come here.
After you know the class, I will sit there.
Yeah. Sure. Please. I mean, you understood the purpose. No? That's what. Don't discuss. Do it on your own. He's in middle. பக்கத்துல வந்துடு ஏன் அங்க நிக்கிற
Dr. Murali, can we join?
Nice. 3:15 it will stop. You have to submit by 3:15. Or 3:20 already?
Just need
3 3:30, huh? 3:30, Abhinav?
sir.
3:30?
I have already set the timer, 15 minutes.
15.
It will ring.
Then it will, you
Yeah.
cannot do anything.
No, I can't do anything.
Yeah, no, before. Close. You give 1 minute warning.
Yeah, I will give 1 minute warning.
Yes.
And then I'll lock responses from there.
Yes.
आपका system open रखो.
Yes. उधर रख दो. The boys are here. Good. Nice. So we have at 3:30, 3:30 the NIF director will come on.
Did it work for you guys there? Your friend? Done? Okay. बोलो. यहाँ से दे. Yeah, I do this very very commonly, so that you focus on reading. I don't know.
Time हो गया?
Sign in to continue. You have to sign in.
I have a mail issue.
Then you have signed in. Now, please. Open here. If you can. Sir, it says 7 minutes odd. 6.5 minutes.
You're done, we'll lock it quickly because
There
there are people who have done.
not able to log in. You don't help him. See, after you have finished your quiz, don't start helping somebody else. I know the answers are open with you. Doesn't mean that you start helping others.
കഴിഞ്ഞ submit ചെയ്താ?
That is not the way of helping anybody. Okay. No, that's also a different thing, but still, I mean, if you seriously want to help them,
In a minute.
clarify their
In 2 minutes we'll close. Bas.
clarify their doubt. I mean, concepts. That is the real help.
Close kar do. One more minute.
Please submit it immediately. I'm locking it. Sir, sir, एक बार open करना होगा आपको.
പറയൂ.
Okay. 5, 4, 3, 2, 1. Lock. Yeah. We have 60. Do we have 60 people here?
Yes. Hey, leave it. Now we move on to the next. We will reveal the answers and all that later. First, let's move on with the program. So, can we get the ഇപ്പൊ എന്താണ്? We are going. So 3:30. Laurent Jaeken, has he come online?
I'm online.
Put the both the chief guests would have come here. വിനീത ഇപ്പൊ NIF-ന്റെ ഡയറക്ടർ വന്നിട്ടുണ്ടാവും ഇപ്പോൾ ഓൺലൈൻ. You change that. First you change that. Let them see what.
The streaming should happen to their laptop. Then we'll be able to see.
Yes. हो गया कौन जीता अपना बंदा मोहम्मद पर सेकंड साला चूतिया नंदन मार गया क्या
अच्छा उसके बाद ना
क्रिस्टीना
4 लोग हैं तो टोटली 6 हो जाएंगे अगर आप थर्ड काउंट करेंगे तो 12 मार्क्स 4 लोगों को
अच्छा ठीक है नॉट बैड कितने 15 में
आउट ऑफ 17
17 ठीक है यस हाय Laurent
Hi, I am online. Can you hear me?
yeah can you put him on hi Laurent
Yeah.
we will have you shortly speaking we are waiting for the chief guest and some activities were going on here so we will have your speech shortly okay
Yeah.
okay just please bear with us I'll call the chief guest if he's available he has come Ranade namaste
Good. Namaste.
Good afternoon nice to have you sir
Thank you.
this is yes
Good afternoon.
yeah good afternoon the other people on the screen are Afsal Manekotodi, Aileen Thatcher, Dr. Ranade, then Dr. Laurent Jaeken. Dr. Aravind Ranade is the director of National Innovation Foundation, our governmental agency, NIF, and it's based in Gujarat. He's originally from Maharashtra. He's basically a astrophysicist, and we will. Can you please put his introduction, Aravind Ranade? Yeah, please, Dr. Aravind Ranade. Can you put that on the? Yes, this is Dr. Aravind Ranade, and he has earned his MSc in Space Sciences from the University of Pune in the year 2000, and his doctoral degree in astronomical spectroscopy from the Inter-University Center for Astronomy and Astrophysics, Pune. And he began his professional journey as a scientist, specializing in astronomy and space science. Space Science Communication at Vigyan Prasar, Department of Science and Technology, Government of India in 2004. Over the next 17 years of service in Vigyan Prasar, then Dr. Ranade has made contributions to scientific education and research through his research papers, articles and books, and audio-video programs. He also worked on frugal innovations, engaging young minds to address teaching and learning as well as local challenges with local solutions. So he's a very nationalist scientist like me. I too call myself a nationalist scientist, though a scientist is the whole world's, but you center it on yourself and your own country first. He is a recipient of several awards and contributions like Leelabai Pitkar Award in 2000, Acharya Shri Award in 2013, Irada Vigyan Puraskar in 2017. He has been a member of the Advisory Council of Academic Boards of various institutions, JNU, NCERT, CSIR, NISCPR, IGUM, Haryana, BAMU, Maharashtra. He is a life member of Astronomical Society of India, the Meteorological Society of India, and the South Asian Meteorological Association. During his career, he held various academic and administrative positions, including National Convener of Vidyarthi Vigyan Manthan, National Science Talent Search, National Coordinator, Vigyan Prasar Network of Science Clubs, registered at Vigyan Prasar. Before joining the National Innovation Foundation as the director, he was the executive director of the Indian National Science Academy, New Delhi, where I first met him in March last year. And then I met him at Bharatiya Vigyan Bhavan, Vigyan Sammelan in Ahmedabad. And that's how I got to know him personally. And that's how I could invite him. And that's, I believe, why he has consented. Can you please put the director and our main speaker today? So I welcome you, Dr. Aravind Ranade, our esteemed NIF director. This is the audience that we have. We have about 70 odd students and scholars and teachers from various parts of India. And we were, for the last 3 days, we were all in this room from morning around 9 to 10 from until evening. So I hope you will have about half an hour with us. At least then we'll have you delivering your speech to our audience. And then we will try to have the thing that our innovation release through you, sir. We will have first we'll give you a broad eye view of what murburn concept is. Professor Laurent Jaeken is on the line. He is making the final speech. So I request you to kindly, if possible, please listen to what Professor Laurent Jaeken has to say about murburn concept and our findings. Thank you so much. Thank you so much for joining. Laurent, can you take over from here, please? Thank you. Will you change the slides there, or do you want me to change, Laurent, here?
Yeah, I'm here. Can you hear me?
Yes, we can hear you.
And can I have the slides, please?
Yes. So, slides konjam podringla? Can you put the slides, please? Yes, we will put the slides. Slides. Oh, okay.
I don't see that well at this moment.
Yes, just 1 second. Yes.
Namaste, dear audience. I started giving the first lecture 2 days ago, giving a broad overview, and now you have had all the details. And I think it is time to recapitulate, but also still add something, because over the days you had listened to different arguments, which were, let's say, the murburn concept. It is thermodynamically sound. All reactions are exothermal. They go on, they go fast. It is kinetically well-founded, and there were many different, astonishing structural details which fit into the concept. Now, there is a last argument, and it is that with classical concepts, you will never be able to understand the origin of life. With the murburn concept, the principle of it, not the details, but the principle of it becomes very simple, actually. So we will first recapitulate what we have learned. Please, next slide. Please, yeah. Here you see that again, the mechanism of the proton pump and the ATP synthase in mitochondria, also in chloroplasts, is quite similar. And down you see the principle of the sodium potassium ATPase in the view of in the classical view. And there is but one thing that one can think about. If it would work like this, the membrane should be very intelligent to run all these things, because actually that is not explained by physical chemistry. So what is the other possibility? It is something very intelligent. So next, please. Next slide. Next slide, please. Yeah. So perhaps we should have a look to how this traditional view came to existence and why it was so much followed all over the world in all textbooks and in all initial courses on cell physiology. The membrane pump theory, that has been clear now that it is fully falsified, and I will give in a few minutes some additional falsifications. Actually, it started with a very famous physiologist, Nobel Prize winner, Archibald Hill. He got a Nobel Prize for his work on the dissociation curve of hemoglobin. And before that year, 1930, there were two schools of thought. Not very much was known about cells, so it was a difficult period, and there was much dispute, and some thought it was a colloid, and some other thought the inside of the cell, its composition, sodium, potassium, other things, it's quite different from the outside. So the difference must be made by the membrane. And Archibald Hill in 1930, he wanted to stop this debate. And he did an experiment, very famous experiment. He investigated the equilibrium distribution or partition coefficient of a small neutral molecule, urea, between a resting muscle cell. It's easy, that's a large cell, so you can easily manipulate it. Between the resting muscle cell and a ringer solution. And he found it was very close to 1. So he took his conclusions. He said, so outside the ringer, that's normal water. So inside the muscle, it must be the same phase. It's also normal cell water. And then there was the next idea, which followed from it. It is, if it is normal water, it is the same phase, then there is no colloid osmotic pressure. It's not a colloid, there is no colloid osmotic pressure. But then, as a consequence, the potassium, which is the most abundant cation in a cell, very high concentration, it should be in solution in order to explain the total osmotic pressure which was measured. It is that otherwise, if it would not be in solution, the pressure would be much, much larger than what it is. So he said the cell is not a colloid. Inside it is a sac filled with a diluted ideal solution, the fluid of it, and it is bordered by a membrane, and the membrane should do everything. And because it is a diluted solution, one can use the laws of Fick for diffusion, which is a statistical law, the law of Nernst for ions, which includes the diffusion component, which is a statistical law, and the law of Van 't Hoff for osmosis, for normal osmosis, which is also a statistical concept. Then in 1940, about the first radioactive tracers became on the market, and it was firmly established that sodium could pass through the membrane and that if you take the law of Nernst for ion gradients, you can calculate this gradient, and the measured values of sodium and potassium differences, they do not correspond. So there is a difference. And so he said there must be pumps in the membrane responsible for this difference. And then in 1957, school actually found this, which protein was responsible. Now, this concept explains in the classic view ion solute distribution, transport processes if they are active or inactive, active or passive, excuse me. It would explain the membrane potential by the fact that the pump would export 3 sodiums in exchange for 2 potassiums stoichiometrically. And if you look to the real data point, then I think about one of the first slides of Professor Manoj, where he had points of data and how you could put different lines through it. And one should be always very careful putting lines and think about which line it could be. Because if you see the data points, you can as well say it is a stochastic process than saying that it is a 3-to-2 stoichiometry. The Chemius modus followed, the cell volume determined only by Van 't Hoff's law, nothing else. And the final important point, let's say that the pump arranges the internal milieu. There were during all the time since 1950 and always some, since the work of Dean, let's say, always some critics, serious, very serious critics. And one was by Schrödinger. And he said, we take again the example of the mitochondrion, from which is what said there were averagely 6 protons in it, how it could work. Let's assume that it is correct, that it works. And then because these 6 protons is statistics, well, it could have been 4, but it could go down to 3. And then the system should start to control the things because it's going the wrong direction. And now it could go again from 3 to 6. That's the point of Schrödinger. You may not use a statistical law with such low numbers. There should be 100 or 1,000, and then you can start statistics. And so he said it's just impossible. You cannot regulate the system. And he was thinking as a physicist, and he said, I know only of one thing which is sufficiently precise to let run something like life. And that is quantum coherence, which we know it exists close to absolute zero in simple physical systems where you have superconduction. He said the only difficult point is we have still to learn how such a thing of quantum coherence could work at ambient temperature. And because of this, probably everybody put that idea aside because they thought it is impossible that it ever could work at ambient temperature. But meanwhile, around 210, 213, it is directly observed on isolated microtubules by some people, among which I think there are, they look to the names, there are some Indian, it's Bandyopadhyay, Ghosh, Sahu, I think they work in Japan, but they directly found that a microtubule works by quantum coherence and is quantum coherent. So Schrödinger was correct. Then there was another important point, and it was by Ling and his some collaborators, his wife and another, and it was in 1973, and he did over the work of Hill. He did the same experiment, and he confirmed that Ring was correct. For urea, the coefficient is 1, but the mistake of Hill was that he was only testing one substance. And from this one, too limited experiment, he made a very general conclusion. So Ling was testing other substances, mostly alcohols and sugars, neutral substances, a large number, and he discovered that the larger the molecule, and it starts from 3 carbon atoms already, the larger the molecule up to sucrose, which is 12 carbon atoms, the coefficient is going down, 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 down. And then the conclusions, which are here still on the slide, they are just the opposite. The conclusion is the cell water is not normal water. It is bound water. It is polarized, oriented water, oriented by the proteins which are there. And then there is colloid osmotic pressure. And that pressure is so great that it almost explains total osmotic pressure. And as a consequence, we have to conclude that potassium is not in solution, but is largely absorbed. There are some in solution, but not many. And so, very big conclusion, one may not use the laws of Fick, of Nernst, and Van Hoff, and the whole membrane theory scrambles. All what is written below, all these ideas, they are wrong. So the thing is firmly falsified. And it is important in science that, as Popper said, science proceeds by eliminating falsified ideas. We have to write things over again from new principles. And one of the big principles, the primary one even, it is a murburn concept. So next slide, please. Next slide. Yeah. So here are then the arguments which Professor Manas was added and which were discussed already. Next slide. Next. Here is the concept itself, and it is really important that in future courses and future textbooks, there are side by side two mechanisms, the mechanism with the active site and the Michaelis-Menten curve, and then this mechanism of the murburn concept, where the reaction takes place in the fluid, not in an active site, and where you get the statistical results. But also, not only the statistical reactions, but because some reactive oxygen species, many even, are negatively charged near the membrane. If from the membrane there comes out an electron and the superoxide is produced, and other negative ROS, you have there electric charge which is generated on that side. And that is just the opposite of chemiosmosis. Here, the charge is generated by the redox complex, by the influid principle of murburn. That's the cause. And as a result of that negative sign there, the proton will enter through the complex 5. If the membrane, if the polarized water near the membrane is depolarized. So then it can enter and control the system, but that's the result of it. And the murburn is the cause, the murburn reaction. In the membrane theory, in the chemiosmosis, the proton flux was the cause, and that proton flux, the generation of the delta psi, was the effect of it. So it's just the opposite. Next slide, please. You have learned through the preceding sessions that the murburn concept has impact at very many different levels. That was also the case for the membrane pump theory. It penetrated in all parts of science. And if you are busy with the part of science which has not directly to do with the things where membrane theory goes about, then it's still in your mind. And you're thinking about making experiments, and this general idea is always running in the background. And you decide to do an experiment this way or another way, just because you have these things in your mind. If you would have the murburn concept in your mind and take it as a possibility, and you should not agree with everything what is said about the murburn, but you should know it exists and take it in your mind, and then you will make experiments and you will be able to better interpret them because you have a broader view and because you have falsified some parts which from now on you can say, yeah, no, not that way. Not, I will no more think about diluted solutions. And you will do better experiments. Now, here down on this slide, you should see the comparison with the combustion chamber of, let's say, a vehicle. What is going in, what is going out, and compare it with murburn. It is somehow similar, you see. And I will make one note by this to discriminate between the stochastic murburn part and the active site mechanism, which also exists. It is you can have the murburn reactions in a flame when you are burning wood. It's very wild. In a combustion chamber of an engine, you can somehow control it, and particularly in an injection motor, because there you inject something at a given time. And in the murburn concept, if you think about the membrane of the mitochondrion or thylakoids or bacteria, it's the same even in the cell membrane. It's the same what the active sites of the enzymes do. They have some kind of decision when the electron will be given. Decision which depends from all the rest of metabolism going on. So I should say that the murburn mechanism, it's particularly comparable with an injection motor. And so this makes a control. Next slide, please. We have learned that many very different proteins can be murzymes. Some are located in a membrane, but like lactate dehydrogenase, it is just a soluble enzyme. And that was a very important session because it showed that even strongly exothermal reaction goes one direction, you cannot do with the same mechanism the other direction. And that's also for complex 5. If you can from ATP hydrolyze it, you will not synthesize it because it is O2 exothermal. You will not go the other direction. Yes, next slide, please. The foregoing picture sets murzymes can be soluble, but they are centered. Many are in lipid membranes because a lipid membrane has the advantage that you can order things better than in a solution, and important molecules can be arranged close to each other, and therefore the lipid membrane was an important step. But the murburn principle, or the essential principle, the stochastic chemistry of it, it happened long before there were lipid membranes. Yeah, next, please. Here it's an overview yet of the different protein functions involved in murburn mechanism at lipid membranes. Next, please. Well, this once more gives the analogy between the fire and the murburn mechanism. I think it's said enough. We can go to the next one. Here you see one special aspect of the murburn, because one of the things produced, it is water in large amounts. And so some mechanisms are just hydraulic, and that's the case for the flagella. It is just a water flow. If there is you know, cells have a cytoskeleton. The cytoskeleton, even it holds everything together. It gives a force which is centered to the interior. And the production of water, it is a force, turgor force, which is want to go outside. And so there is much pressure on it. And so I can say that in a cell, there are even small narrow channels and clefts where there is not, you have normal water, you have polarized water, which is much more viscous than normal water. And you have also hyperfluid water. For instance, at two places where polarized water meets with opposite direction of their polarity, there is an intermediate, very narrow, and there it is hyperfluid. And also in the case of the flagella, this channel is so narrow that what goes through is hyperfluid. It is really a jet, like here with a rocket. And that's a simple system. You have only to produce and have the narrow channel. That's all. And because the flagella is asymmetrically built, you translate this motion of the fluid into a wave, not a rotation. You do it in a wave, and every fish knows that you can swim with it, with a wave. Okay, next, please. Next slide, please. And here I come to the last argument for murburn, and it is the early origin. And you have a time axis, and it starts at the bottom with some simple building blocks which are present. And there is one important among this, and this is iron, which was abundantly present very early on the Earth. And there was something else which was abundantly present very early, and it was UV radiation. And iron is very sensible to UV. And during the night, the iron was reduced to its state of 2+. And during the day, it became photo-oxidized to its state 3+. And then this can react, this 3+ can react with water and generate dross from it. So this means that there was even oxygen in this anaerobic million, but very, very small quantities. Every time that an electron was donated, it could be produced. And also now in anaerobic bacteria, strictly anaerobes, there is continuously small amounts of dross and oxygen produced, and that's why these organisms have a super oxide dismutase. So the murburn also works in the strict anaerobes, and it can work with dross, but it can also work with a reactive nitrogen species and can work with a reactive sulfur species. In anaerobes, the other are important. So if you have oxygen, it means if you look to the redox potential, you can already use the whole span of very reduced to very oxidized. And you have already the primordium of catabolism and anabolism, which exists already. And you can start with all reactivities. So the murburn was first, and one of the things it did, the ROS, it was to attack the methane and start to oxidize it. And you can have some carbon radicals formed with which you can have lengthening of the chain with two carbon atoms, three carbon atoms in the beginning, perhaps four. And you can introduce in this hydrocarbons, you can introduce oxygen with it, making hydroxy acids, keto acids, carboxy acids, and if there was ammonia, so you can make amino acids. So this means without the dross, you would never have had amino acids. Let's say you would have life. So the dross is really at the beginning. Then there is a second important step in the early evolution. That is that even with a very limited amount of amino acids, maybe with take just the most abundant. It is glycine, proline, aspartate, glutamate, alanine, and valine. Two are hydrophobic, the other are hydrophilic. You can make something like resembling a gelatin gel, where there is also very much glycine and proline, and it's a hydrogel, and it is made because I take even if there are small change, because it was done in an experiment with only 14 peptide units, you can already have made coacervate. And the coacervate, what it means, that you go from the size of the molecules, which is nanometers, you go to the size of a cell, which is micrometers. That's the first effect. But there is a second effect. You go from normal water to polarized water. And even again, when two polarized regions meet each other and there is a narrow space in between where there is hyperfluid water. So the very special waters are there. And the polarized water, it is very odd, you have already coherence. And with these coacervates, you have, and with the murburn inside, I should say, the murburn and the coacervate, it goes very well together because, for instance, with the murburn phosphorylation, you need a low water activity. And then you can make ATP. Well, just inside the coacervate, there was low water activity. So I do not say that there was no formation of ATP outside the coacervates, but inside it certainly worked at a higher pace. And then at a moment, much later, there came active centers and they started the injection motor of it. And much later, there came RNA and DNA. And then things no longer, before everything was real-time chemistry, from that on, systems could be better reproduced. A reproduction and Darwinian evolution could start. Due to the cyanobacteria, there came first by photosynthesis hydrogen sulfide and later oxygen, the oxygenic photosynthesis, and oxygen accumulated and we got aerobic cells. That's this picture. So you see, this is something easy to come to existence, relatively easy to come to existence. To make a Boyer complex 5, no. How you could ever make that in small evolutionary steps, because it should be perfect before it can work, and even then it doesn't work. Next, please. Next slide, please. here I want once more to stress that the murburn concept is the oldest, the most basic concept, and then the other things came later. At some time there was a cell with a membrane. Okay, then we can speak, from then on we can speak of a cell, and there is a central dogma, and once the central dogma is installed, reproduction and standardization of the thing can go on. But perhaps just tell something about the murburn concept and the co-observation. These are extremely, you can trust on these things. Because with many very different oligopeptides, which initially institute different sequences, there will, with the 6 amino acids, there will always some parts which are sticky, and there will some parts which are exposed to the water. And in a polarized water, sodium is not very well soluble, so you will have a low concentration. And with the aspartate and the glutamate, you will bind the potassium, and you have your sodium-potassium distribution just as an equilibrium. So I mean, among the different co-observations which originated, many would have been good. It is like Darwinian evolution now. You should not think the survival of the fittest, because the survival of the fittest is the top of the Fujiyama in Japan. There are all, it's the fittest, they are not only the athletes who are going to the Olympics. All of us are fit, sufficiently fit. There are many possibilities which all work. And also with the co-observator and the murburn, there are many possibilities that works. So it is really a good workable concept. Next, please. Next slide. Yeah, here are the examples of the many possibilities which do not work, and the alternatives of the murburn which were shown to be a possible alternative. Yes, we go to the next slide, please. Oxygen, I can only tell one thing. Before the investigations and discoveries of Professor Manoj, oxygen was the forgotten molecule. And before the work of Ling, water was the forgotten molecule, which was not built. And also the idea of coherence was the forgotten idea. I was once, more than 20 years ago, in the library, and I was looking in the shelves of biochemistry and cell biology, looking for the word coherence, I didn't find. And in the other side, there was physics, and I went there, and I looked for the coherence, and just I suddenly found a lot of physicists who were working on life processes, and they were telling about coherence, and that's nice stuff. So coherence is forgotten, oxygen was forgotten, water was forgotten. Therefore, it didn't work. These are the 3 main important points. Next, please. Next slide, please. Yeah, now we come to the philosophy, with which Professor Manoj started. And it is the task now of the new generation to finally get rid of a consensus view. And the alternatives, they develop very slowly. There are some people who start that there is thinking that there is something wrong, like Schrödinger did. like Lingnick, like Professor Manoj did. But they build new models and they are discarded, they are marginalized. And I will say in Western science, there are other ideas which are marginalized, like, for instance, the key principle, prana, and others. They are marginalized. Yet these are things, they exist and they're worth to be studied. And then if we take the new cell physiology, which is now, we will start to understand, because I will tell one thing more. One function of Ross, which was not yet explained. It is that every reaction in chemistry, every inductive effect in a reaction, there is a displacement of electrons along a binding axis. And this means that some electromagnetic radiation is either emitted or absorbed. And every detail what happens in our material body is found back in the radiation, the electromagnetic radiation which is emitted and absorbed. It is there, the same information, working at almost light speed. And we have a halo around us which full of information. It extends out of material body, but which is ethereal and which can be measured by sensitive apparatus. And that's also a marginalized aspect of life sciences, but it is there. It is like a hologram, which is important to coordinate everything. And already these first co-surveys, they had it, because it is just basic chemistry, they had it. But as order increased over evolutionary times, the system and the hologram became always more complex and complex. And it is the thing which is able to coordinate everything. And I heard part of the discussion after the last session, which was, I think, going on about homeopathy. But one of a Nobel Prize winner, Montagnier, who got a Nobel Prize for his work on the AIDS virus, was one actor, and the other actor was from Milan in Italy, Del Giudice. And he derived that water consists of polarized water and non-polarized water. And in normal water, the polarized form is a minority, but in cells, the polarized is a minority. And they can exchange individual molecules, but on its own, the polarized system is very, very, very stable. And he took the two work together, one knowing to calculate frequencies, and the other a fine virologist, and they took the DNA from the virus, isolated, diluted like it was done in homeopathy, and then diluted so that one is absolutely sure that there are no molecules no more in it. They went, I think, 10 ranges further than when it should have already no more molecules statistically. And then it was repeated many times. And one should do that for such important experiment. And they had this tube with no longer DNA in it. And from this, they added the DNA polymerase and the primers and the nucleotides. And they got again the DNA from the virus with only a not completely correct, but only 2% mistakes. So it means that the information which is in the DNA is surrounded by water, by polarized water, particularly in the major groove. And that contains all the information which is also in the basis sequence. So this may be not a proof for homeopathy, but it anyhow says that things are much more complex than the traditional view. So with this, I would tell you that there is still very, 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 very much to discover on very important things. Just keep an open mind. And now I will let the last slides for Professor Manoj.
Can you hear me?
Yep.
Yeah. Thank you, Lauren. Thank you, Professor Laurent Jaeken. I would just quickly, our chief guest is waiting, and he's a busy man. He's a director. So I'll quickly browse over the next few slides. Please bear with me, Dr. Ranade. So this is the message. Can you you can put him in background if because I can. Okay, no problem. No problem. What are the essential facets of life? These are the take-home messages. Please pay attention. The take-home message is what is the most, see, science answers questions, right? Question, answer. Question, what is the most salient feature of life? A living being has the ability to preserve itself by commissioning non-spontaneous actions. What is the most essential requisite for the above? A simple powering modality interconnected with coherence, homeostasis, and response principles. So there are other definitions also. Replicate, mutate, catalyze. Horowitz example, that's good also. Does the cell theory or central dogma provide tangible, adequate means for achieving the above non-intelligently and at short time scales? No. Murburn concept addresses the above need and complements the cell theory and central dogma as yet another fundamental principle of life. Please see that Murburn concept does not negate it, just complements it. So what is the etiology expansion of Murburn concept? It stems from mured, closed, confined burning or a redox process. A concept is a forthrightly derived idea or a set of ideas starting from known facts. So how is Murburn concept relevant to life? Murburn concept conceives a cell as a simple chemical engine performing useful work via the principle of effective charge separation brought about by the obligatory movement of diffusible reactive species. Why is Murburn concept a fundamental principle of life? Murburn concept provides a non-intelligent and stochastic framework of interactions between diverse molecules and ions that constitute the cell. In doing so, it enables a simple explanation for powering, homeostasis, coherence, signal transductions, electromechanical activities, etc. So, what is the salient difference between the classical and Murburn purviews? Classical perceptions primarily deem diffusively reactive species as toxic waste products, whereas Murburn concept school deems DRS as quintessential elixirs of life. Does Murburn concept oppose or negate classical perceptions? Murburn concept is a larger set of ideas including several aspects of the classical views, incorporating later known facts and improving upon earlier prevailing stigmatic aesthetic notions. In quantitative perspectives, what is the impact of Murburn concept? Classical perceptions had purely deterministic schemes to account for metabolic or physiological outcomes, thereby mandating integral stoichiometry, whereas Murburn concept accommodates stochastic events, thereby explaining non-integral stoichiometry and atypical kinetics dose responses in cellular processes as contributors in routine function. So this is a cartoon strip I usually put in my scientific lectures at the fag end of it. Bill Watterson is my favorite cartoonist. This is Calvin and his imaginative buddy in the stuffed tiger, Hobbes. Hobbes is also a philosopher, if you know that. So you always, if you're a scientist, you should have the childlike mentality. If you have lost it, you're not a scientist. You're not a scientist. If you just take things, if you don't ask, what is that? What is this? Why? How? If you don't doubt, if you don't question, if you don't challenge, there's no point. So here it says, it's a magical world, Hobbes, old buddy. Let's go exploring. That's what Professor Jaeken said in the last, sentence. You need to keep an open mind, explorative mind, inquisitive mind, questioning mind, challenging mind. That doesn't mean that you become ungrateful or disrespectful. You might be opposing your seniors, but do it with a decent way, right? That's how new things are coming to the society. The older people, they kind of maintain the society for you. Always understand that. So with that, I come to the end of this scientific part. And our chief guest today, can you put him on, please? Our chief guest, please. Our chief guest. Please bring him on, please. Yes, Professor Ranade, if you see his CV, he has been working with the society. All that we see, the structures here, without the society, science, zero. Without a darbar, vidwan, zero. Do you understand? And the darbar is made by the people. So our chief guest today, he is a scientist as well as a person who has always worked with people. You see his CV, I read it out. That is what. He has worked with people. And now, Ranade ji, please address our gathering. Please, thank you so much.
Thank you, Professor Murli Manoj ji. First of all, I really compliment and I congratulate Professor Laurent Jaeken, who very beautifully, gave the idea about the murburn concept and the importance of the new dimension that has been coming up in the biological sciences. And I really appreciate, all the audience, since my CV has already been known to you, the kind of background that I belong to. However, when it comes to the life sciences, this is the reality, and we all are so much close to the biological sciences because every breath that we take, make us alive, is only because of the biological sciences. Though I have only up to the I can say up to 10+2 level of biological sciences with me. But it is very interesting to understand and realize the new dimension that has come up. And I really compliment for Murli Manoj. And to address to all of you, it really gives me a great pleasure to address the symposia at Amrita University, addressing how the cell work at the fundamental level. As a director of NIF, which is a National Innovation Foundation at Gandhinagar, which is the autonomous institute working for the grassroots innovation and innovators at the Gandhinagar under the Department of Science and Technology. We really look forward for such new initiatives and activities in our country because in the contemporary sciences, the new ideas and new initiatives and new innovations is the ultimate kind of solutions to the problems that we all as a humanity look forward. I remember first time I met Professor Murli Manoj ji at Delhi when I was the executive director at INSA. When he was, one of the senior advisor from the DST, asked him to come and meet us because academy had an excellent, intellectual kind of a group working for the science, especially when it comes to the INSA. It is a highly biological sciences who are the prime pinnacles of the biological sciences who work in the country as well as on the international forum as well. And I very candidly remembers the
Ranade ji, can you hear us? Hello, we have lost you. Ranade ji, Aravind ji. Can you please disconnect and perhaps come back?
idea And one of the example that we just have seen, the way our expert was
Dr.
speaking
Ranade,
about.
Eight minute. We lost you for about 20 seconds. Can you just give us your message? 20 seconds, we lost you. Yeah, you were talking about.
I see. So sorry.
Yeah, we lost you for about 20 seconds. The screen went, stuck, and we couldn't get your audio for about 20 seconds, 20 seconds. Yeah.
Okay, okay.
Yeah.
I don't know from which particular aspect that was lost, but I was trying to explain and then speak to the audience, especially the youngsters who are trying to work with this new concept of murburn and how this is going to be changed the way that the biological sciences look forward, because the 21st century is full of the concept and the new dimensions for the biological sciences that I could see, and I was trying to explain them how we met. And I do, very candidly remembers that your visit to the Indian National Science Academy when I was executive director there, and being an academy which is a highly accomplished members of the academy who are at the national and international repute, who has come up with the very dignified work, which actually helping the new sciences to bring forward the ideas and concept. And I really remember the idea that you shared as a modern concept, how the new dimensions are going to add up in the biological sciences. And the same thing I was trying to explain the audience that we again met during the Bharatiya Vigyan Sammelan at Ahmedabad, wherein you again came and spoke to the audience about the same concept, which was, and then in fact, as far as this symposium is concerned, this murburn concept is altogether a focal theme for the current symposium. And by listening to the Professor Laurent and the kind of the aspect that I have gone through with the brochures that you have shared with us, it is really it really gives me an idea that it is a significant pursuit, and and you have published a good amount of papers in the reputed journals and at the national and international level, and it has got a really impressive ideas. Now, and I really wish that my youngsters, that Professor Manoj will inform me that with this artificial intelligence, which is on in the department, is working on it and trying to come up with the new database in terms of the murburn enzymes, in terms of the new ideas that is coming up. And it really gives a great pleasure, and understands that this enzyme's functionality and the way that it is being coming up as a first database for the global community to freely access and avail the informations on the new frontier. And as an official governmental agency and that aids and apps such ventures, it gives me an immense pleasure to inaugurate and commissions the same for the public usage. I commend this Amrita Vidyapitam and the School of Artificial Intelligence for these achievements, and the hope that Professor Murli Manoj and his team, who is the very youngster, dynamic people who are trying to take this new concept and come up with the innovative endeavors. And I hope that this particular database is going to serve for the many, I would say, the community of experts across the globe for trying to come up with the new dimensions and the way that the biological sciences will look forward. I also congratulate Murliji for this hypothesis and creating a new database for predicting the enzymes and following the mechanism. This is a new dimensions to all to the newcomers in terms of the needs of the time. And I would say that I really got more knowledge when you put on some of the questions and answers, which has given us the fair enough idea for the newcomers how the science should be taken up when the new concepts have been coming up, how one has to actually look into this, and how one can actually try their efforts to make understanding the natures in the best possible way. In fact, Professor Laurent was also explaining how this is going to help in terms of the homeopathy, in terms of the allopathy, in the days for the modern sciences. With this, I really, thank to you and the organizers for giving this opportunity to speak to all of you. And I look forward for the new dimensions and the new outcome of this work that you all are carrying at Amrita Vishwavidyalaya. Dhanyawad. Thank you so much. Thank you, everyone.
Thank you so much. राणादे जी, एक मिनट आपसे थोड़ा 1-2 मिनट और लेना चाहूंगा. I would like to introduce you to the team that, the main actors of the team. They are the students. Please come. Preetham, Omkar, and Vineet, our colleague. We have this team that has Amrit, sorry, my bad. Amrit and Omkar and Vineet. Vineet is the faculty member who has been guiding them. We have also other colleagues who have guided them. So these two, they'll just show you what it looks like. So now it is connected. So they'll just show you what it looks like so that you will see what it does basically. Yeah. 1 second. Yeah. So what it does is basically we have unleashed a new functionality called murburn functionality. So we are using AI methods to predict whether this particular protein or set of proteins would be, so you can retrieve some PDB, that is a protein structure or information about it from the web, and just put it into this, and it will predict whether it is a murzyme or not. So currently, what's the kind of accuracy that we are getting on it? 85. So would you want to speak to our senior members and the audience, Vineet and students? The stage is yours. Have the mic, please. Mic is there. Is it on? No, it's not on.
Hello.
Yes. Come here and show them whatever. If you want to. Yeah. You connected. Camera. Yeah. You need to come to the camera if you want. Otherwise, it's taking them. You come here, it will come. Yeah. Just show them a demonstration so that it becomes,
So a very good afternoon. I hope it's audible. This is Amrit and I have Omkar with me, and Vineet is there, our guide. So this web app was created to make the research on wherein concept a bit more lightweighted and easy, because collecting data is the most important part and making models is the toughest part in it. So we made a central hub where you have all the data points mostly. If you don't have, we will scroll through the internet and get you that. So we have a custom made API retriever for it. So firstly, I will explain you the concept. The first concept is, or to put it simple, the first feature is the feature DB, where you can type any title or keywords of a PDB file or the murburn file which you are searching for. So I will type copper amine, and then you will get the particular PDB file that is related to the keyword which we typed in. So if you see, the title says copper amine oxidase from Arthrobacter gloriformis, it just goes on like that. So if you leave it empty, all the first 50 entries are shown, and then you can even search it based on the keywords you have. For instance, if I type oxygen storage,
I mean, that is storage.
It's a scientific key term behind it.
Yeah.
So as I told you, you will get entries based on it with oxygen storage in it, and then you can even search for murzymes and non-murzymes, the normal thing, MURZYME. So you can just search for it and you'll get all the murzyme entries. And then if you go for non-murzymes, no, that's how it works. So that is the first feature, and then we are moving to the most important feature. This is our non-murzyme and murzyme classification thing. So we had around 600 data points, and we trained a supervised classification model called as SVM, singular value decomposition. It ran on RBF kernel. So honestly, we took 3 to 4 models as entry members. It was logistic regression, SVM on RBF kernel, and then random forest, and then another model. So this was the best performing one with 85% accuracy. So what you have to do is you can go to the app and then download a Murzyme file that you want to. So I'm downloading this. So when you download it, your PDB file gets saved, 1QRD. Now what you can do is you can go to our model prediction thing and then browse files. So it's very simple to use. You have to take a PDB file and then upload it. So when you upload it, yeah. So when you upload it, the file says whether it's a Murzyme or a non-Murzyme. So the file which I uploaded, it's 1LOP. You can check it on the database. It's a non-Murzyme molecule. These are the details for it. Title, crystal, structural analysis of the complex between, it just goes on like that. So the approach which we took here is that when you have a PDB file, we are extracting 3 to 4 features like the title and the keywords and then the actual name of it. Then what we do is we are using a transformer-based embedding. It's a deep learning model. So it is responsible for converting the text we have to vectors. So they hold the semantic representation of it. Semantic in sense, the actual meaning of it. And then we are storing it in a database and then we pass it to our model. It's of 378 features or 378 entries to put it simple. And now the other two features, the PDB ID search and plots will be explained by Omkar. Thank you.
So this is Omkar, and then third feature is about that to search PDB file or to get a PDF file to retrieve the PDF file from the RCSB website. So what you have to do is here you have to just enter that any PDB ID. For instance, let's take one A to B. You just need to be very particular. Means it's a case sensitive. You can't just give one small a and two small b. It doesn't work like that. So you have to be very particular about this one. And when you just try using a API, it's running. Using a API search, we have just this one. This is the third feature. So we got that file. This will be done. Thanks. So it shows that the it matches with the PDB ID, and we got that file. So you can whatever you can download that PDF file if you want. Yes, it's available. This one. Ah, yes, this is the one. So this is what I, just now I have downloaded it from the internet. Yes. So this is got about that feature, and this the last feature is about that plot thing. So as it is very high, it's a very high dimension, so we can't visualize it. So we just using a technique of PCA and TNAC from the FASTA data points. We just converted it to 2D so that we can get more insights. We can get more insights, so we can dive further for the future thing. So we have four features here. So it's the light blue color shows it is a non-Murzyme thing, and the dark blue color shows it is a Murzyme thing. So we have four features, and based on that we have plotted a scatter plot thing, and these both are the PCR for the fast data points, and down here we have just other PDB data files. This is the PCA, and this is on the TNSC. So that's thank you.
Vinith, you want to say something? No? Okay. Thank you so much, Ranade ji, Aravind ji, for your attention and for gracing this occasion. So we will, I think if you're busy, if you're not busy, we have function going on, but I think you might have to go on. Thank you so much. Hopefully you can come to our institution someday when you are you were about to come, but then the minister called you over to Delhi, and that's the reason you could not make it. Hopefully you can come to our institution and grace our campus someday. Thank you so much.
Thank you, Murliji, and I really compliments to Omkar and his team for really bringing up a wonderful database. I look forward to have a very fruitful, outcomes of the entire AI-based work that the team is doing, and I really look forward to have a this concept to be not only, by the community, but as a practitioner, the good amount of outcome will be coming up with this new concept in the biological sciences and especially in the cell sciences. And I look forward to have an in-person meeting at the Amrita Vishwavidyalaya very soon. Thank you so much. Thank you, everyone.
Thank you. Thank you. Thank you. Thank you. Thank you. So, who are the other people there? Aileen Thatcher, Hiroisa Tamagawa. All these people are my well-wishers.
And it's me.
Yeah,
Myself
yeah.
also there.
Yeah, Afsal, that's what I was going to mention. Afsal Maneko Thadi is my another soul friend, student, my well-wisher. He is currently he's a physicist. He's in Oman. Then Aileen Thatcher, you might have known her and seen her. I don't know if she is listening. It's early morning there.
Yes, I'm here.
Oh, wonderful. Nice. Hello, Aileen. Would you want to talk to them? I'll just introduce Hiroiza also. Professor Hiroiza Tamagawa is from Gifu University, Japan. He is actually a mechanical engineer who is into macromolecular chemistry and all that. So he had actually made that. I showed you the equation, Tamagawa equation, which was exactly like Hodgkin-Huxley equation. So Hiro, are you there? Hello, can you say hello?
Can you hear me? Can you hear me? No?
Yeah. Yes, hello. Hello.
Hello? Hello?
Hi, hello.
Hi, nice to see you again. Nice
to see you.
Nice to see you again. Sorry, because of my son's issue, medical issue, I'm sorry. I couldn't participate. I'm sorry.
Yeah. Yesterday he was supposed to give a talk, but his son had a medical emergency. So would you like to say something to the audience?
Actually, I'm weak in English, very difficult to express my mind. But I like to say congratulations to all of you, not only to all of you. Congratulations! This is a great opportunity for the murburn concept and new physiology. I'm really happy. Yeah, thank you.
Thank you, Hiro. Aileen, would you like to say something?
Sure. Let's see. Is it going to come on? There we go.
Hi.
Yeah, I've been up all night.
I know.
3 days, no sleep.
I'm sorry.
So
Yeah,
I'm violating my own rule. No, this has been a fantastic meeting. It's been it seems like both a deep dive in some ways and surfing over something so fast. I think everybody should review the collection of information that you've had and really thoroughly go through the papers and digest this. As an immunologist, which Murli has mentioned a few times here, it's like my worldview is very systems biology oriented. Unfortunately, our knowledge of immunology is most deeply centered around mice and man, which is really not the representation of the diversity of life. But the principles that are contained here are the core principles and the foundation of immunology for all organisms, and that's in all kingdoms. That includes fungi and plants. They do have immune systems, and their core principles are the same as ours. And it really revolves around ROS in so many ways. And not just the ones that were highlighted mostly during the last 3 days, but areas where I've spent a lot of time was in hypochlorous acid and to a certain extent hypochromous acid, which hypochlorous acid, to give you an idea of how reactive it is, it's 1,000 times more reactive than hypochlorite, common bleach. Much more effective in so many ways. And so it takes very, very, very little, and our cells generate it all the time. And hypochromous acid is 1,000 times stronger than hypochlorous acid. So just imagine. So studying these things are challenging because they are very reactive, very fast, very small amounts. So looking at it is difficult, but I think it's important to start bringing our focus to different levels to have a greater understanding of how and where things work. And the other aspect I think that I don't think has been pointed out as much, but it should be, is that reactive oxygen species have been sort of created and demonized as sort of the bad guys. And so much of our pharmacology and everything is to get rid of them, like NSAIDs and so forth. Let's interrupt these processes and chains like uraconic acid pathways. Let's, get rid of these nasty things that are, causing inflammation and so forth. But it's these very things that are making life possible. It's how we can control infections. It's how we can heal. It's how we can remodel tissues and so on. So I've already gone on too far. But in other words, this 3-day symposium is just a toe dip in the giant ocean of this enormous subject. And it's very exciting. And congratulations to Murali and his whole team. This has been a really, real great effort. And everybody should applaud them. Thank you.
Thank you. Thank you, Ed. Thank you. Thank you. Dr. Aileen Thatcher was a professor in Sonoma University in California, and she is now associated with Amrita, our mission and our university. And we met when she came here, and we have been hooked ever since then. Nice. Yeah, thank you. Afzal, would you want to say something, please? Quickly. Can you hear me, Afzal? Can you come?
Yeah, I can hear you, but unfortunately, I cannot open my video. My video has some issues.
Okay, no problem.
I just want, first of all, I would like to thank Dr. Murali and congratulate him on holding such a wonderful activity. And I see a lot of young kids actually, which I know that your key focus all of your life is. And actually, when I heard a story, I think it was Abhijit saying his story when he met you in a campus in VIT somewhere. Is that Abhijit? I forgot the name. Who was it? I also met Dr. Murali like that. And since then, we have been interacting all sort of discussions throughout our life. Now, regarding this conference or symposium, I would say it has been a milestone event in murburn history, and I hope it gonna be.
Afzal, we have lost you. Hello.
Can you hear me now?
Yeah.
I'm not sure actually. I was saying that this event or this symposium will be a milestone event in the murburn history, because this has attracted a lot of students here. And I was looking at, actually I was with you 3 days along the way. Sometime I go and back, but I was hearing very active discussions, debates, and primarily students are interested in it, which I believe they will take the murburn concept far ahead in future. And I also see some decision makers also coming to the conference and commenting their own support for you. And I hope we will make murburn as the next challenge and next future. And I wish you all the best. Thank you.
Thank you. Thank you. So, Lauren, Hiro, Aileen, Afzal, thank you so much. You've been a pillar of support all through this journey. And it's this principle. You build on what you have and what you know. These are the people, they have been supporting. So now, thank you so much. We are moving on to the young people who have been here waiting for the results of the quiz, right? Can we announce the... Huh? You got it? Who released that? You got you can see that? So who's the winner? They don't know. Oh, How many questions were there? 70. Lovely. So how much do you think would be the highest marks? 70. It was not bad, right? 14 was the highest, right? So 14. Who's got 14? Yay! So uh, so there goes your prize money. नहीं नहीं टॉपर अभी मेरा 500 500 कितना लेंगे हम नहीं नहीं एक मिनट हाँ वो तो ठीक है फर्स्ट प्राइज इज 500 सेकंड प्राइज नो सेकंड प्राइज नहीं सेकंड प्राइज है सेकंड प्राइज किसका है यस हैव अ लुक अरे कौन था किस नेक्स्ट इज 13 मार्क्स खाना है ना हु गॉट 13 Who got 13? Dayanidhi is left. हाँ ठीक है that's good. That guy I thought he was a fraud. My bad. No, he was attentive. I thought he came here for different reasons. I thought he came here, bird watching kind of a thing. Yeah, somebody was telling me he was collecting phone numbers of girls and all that. Yeah. Yeah, but it's a surprise. You never know. You never know. So there are a lot many 12s, right? How many 12s? Can the 12s please stand? Please raise your hand. 12s. Nice, nice, nice. Good, good, good. So you have been attentive. Nice. You got some funda. So there were more. I think they have left. Four. Yeah. 1, 2, 3, 3. One more. Huh? Yeah. Nice. So maybe from the team. Yes. So I think 50 rupees. It goes like that. So 50 rupees for the two of you. Yes. You're receiving it from my hand. I think it's important. The originator of murburn concept. 50 rupees de de yaar. 50.
You never changed, Dr. Murali.
No, 1 minute. Yeah. So you got 50 bucks. Give me back. Yes. He's taken 100 and he has said thank you and given me his hand. So one more 100 rupees, please. No, 100 rupees. Yeah, 100 rupees. So 200 de de. 200 de de. Unko de denge. So give me that 100 rupees back. No. So this is your 100. Thank you. I'll give you the 200 later. Yes, wonderful. So what more do you want in this now certificate distribution, right? That's an official formality that we have to. I am the chief guest now. Nobody else is left. We have the co-convener also. Let's give away the certificates. Please, I invite Vineet. Thank you so much. So we had other participants also in this. We have had Muthu. He has been very supportive. He has been here. We have all these things do not visualize without support people. Muthu has been with us very. Please put your hands together. I call him Muthu, but he is Dr. Muthu Krishnan. He has a PhD in chemistry, particularly synthesis of very sophisticated kind of molecules. So Sampath, please come. I want you to. Sampath also has been, I wanted to go there, go here. He was helping. I wanted this and that. He was helping. He's our technical staff. So, yes. And before we started doing this, I want to put a political statement also. It's sad that this institution has 80 research scholars in chemistry and physics. 80. We have had only 2 registered. Do you think it's a good thing? No. Do you think this symposium was good? Yes. So thank you for coming. Thank you, Arun. All right. Thank you, Rajesh, for coming. Though you missed some part, but you registered. Thank you. I appreciate that. When somebody makes an effort in your own institution, when your own institution, students, faculty, research scholars don't participate, is it good? No. What does it show? There's a matter of serious concern. And when it is not good. So that I wanted to say. So let us now start with the official formalities, please. Yes. Can we read out the names and can we have them come over? Please, Vineet. दोनों मिलके देते हैं. Let's go. Read out the names and with that, after the certificates are given, I think we can stand and talk, or whoever wants to go can go. The time is done, right? Yes. Let's you just yes. Give him a mic. And I wanted to before I finish, before we started with this, because my Mr. Joshi, Prabhakaran, wonderful hosting. Please put a hand for them. Wonderful people. Please come, just so that you see, getting all these things organized, it's not easy. So thank you so much. This is Mr. Prabhakaran. Please, Joshi, please come. I want you to get into this range. I want this. Please come here. I want you to be registered as I want it. I know what I'm doing. Yes, please. So this is important. They help me. For example, one of my guests, his mother wanted food. He ran. At that moment, all that touches you seriously. Thank you so much. Thank you. I appreciate it. So my management, my Soman sir, my colleagues, thank you so much. So there is usually official thanks and all that, but it's not scripted. Thank you so much for coming. So now let's do the certificates and then we'll do that. Yes. Sampath nahi pudichuko manana. Santosh. Yes, Vineet, come. Yeah. Ibdi. Vignesh. Kavin. Kavin. Vijay Mitchell. Chittu kedachada. It's okay. Abhishek. Look there. Session. என்ன போல சொல் ஃபியூச்சர்லையா இப்பவே பண்ணிக்கோ டிரான்ஸ்பிளான்ட் பண்ணிக்கோ வேணும்னா நல்லா பிரச்சனையும் இருக்காது சௌந்தர்யா ஆஞ்சலின் கிருபா Kritika, Yuvashri, Balamira, Jennifer. You should be willing to face. Okay? Hello. Balamira.
Thank you, sir.
This is what happens if you're not prompt. Somebody else will take away what's yours.
Gauri.
Yes. Got it? Yeah, quick.
Jayapriya. Arti. Arti. Genova Mercy. Beulah Rose. Aruna. Hema Priya. Remy.
Hi. Yeah, yeah. Thank you for participating. Hi.
Sri Mugila.
Did you attempt? Yeah, okay, good.
Thank you, sir.
Yeah.
Saumya. Sanjay. Logeshwaran.
So you're Sanjay. Yeah, we talked over the phone.
yeah. Monica Sri.
Good. Yes. You didn't screw up my phone, did you?
No. Reshma. Joyce Priscilla. Hema Ranjini. Indumadi. Sridhar.
Thank you, sir. Thank you, sir.
Ardra.
Hey. Chalo.
Christina. Akshada. Akshada.
People were paying attention. It was not bad, no? They got
Sri Lakshmi.
14 kali high.
Aditya.
Seri okay. Not bad, no?
Sneeja. Kavya.
Guys are.
You're Aditya.
Kavya KL University.
Yes.
Yamini.
Yes.
Sai Deepika. Vijay Bhargav. Mangal. Priyam.
Yes, sir.
Sonam Kumari.
चला गया प्रसाद
नहीं वो आएंगे सर
हाँ
Srikaram Adarsh.
एक और ले लो नहीं नहीं फोटो ठीक नहीं आया
Okay.
Yeah.
Thank you, sir.
He is my student's student. She couldn't come. He is my student's student.
Srikaram Abhay. Bhupalan. Bhupalan. Naveen Raj, Jayasudha, Sweta, Harihara Sudan,
Good.
Manimaran sir,
Yeah,
Sridhar,
yeah, you can, if you want, you can have the snacks and all that, no?
Those who have got the certificates can go.
You can go and then come back if you want. We are here. We are not going. Yeah. Yes.
Yes.
oh Sridhar already கொடுத்தாச்சு. Nemadullah,
എന്താണ് snack? What is a snack? போண்டாவா நல்லா இருக்குமா? ஏய் ஏய் சட்னி இருக்கா? டேய்
Yogita,
Thank you, sir.
Vaishnavi,
Jiddu Vaishnavi, Jiddu Vaishnavi.
Jiddu Vaishnavi,
Jiddu Krishnamurthy, Jiddu Vaishnavi. So nice to have you. Seriously.
Mohammed, Maniyar, Mohammed,
Certificate of merit we have to give to some. Hey, certificate of
sir,
merit for
please.
the people. Sanjay, has he gone? Sanjay, stay. There is certificate of merit for you. There is certificate of merit for you.
Dr. Pradeep,
And the two girls also.
Dr.
You have
Pradeep.
certificate of merit. Wait.
Congrats, sir. Wish you very, very more and more success in the life.
Karthik,
And
Karthik RS,
Yes. I'm already at the top. I feel I have got the love of my people. What do you
Snigdha,
want?
Dr. Snigdha,
always, always. Thank you, sir.
Wonderful. Keep the spirit up.
Yes,
I
sir.
love you.
Always, always.
Yes. But you understood when I cut you short, no? We had to get with the program. Yes. Nice. Karthi, you saved our institution's pride. Seriously, our participation is important. And you showed that, when you came regularly, that really means something to me.
Ramachandra Karthik,
We should.
Niranjana Prashant,
It was so nice having this conference.
One more.
Raj Mahalingam,
Rowdy. Raj, he doesn't want. He's
Okay.
senior. He doesn't want.
Arun,
வராதவங்களுக்கெல்லாம் எடுத்து தனியா வச்சிட்டேன்.
யாரு?
நம்ம எஸ்ஆர்எம் சார் இதுல அமிர்தா
Yeah.
ராஜேஷ் எல்லாம் வரல.
Okay.
ஆனா சர்டிபிகேட் போட்டுருக்கோம்.
Yeah, okay. யாரு இருக்கானுங்க? ஏய் ரஜீஷ். வா வா. Yeah. Yes, sorry. Rajesh இல்ல, Rajesh. Yeah, it's okay. Ba. ഒരു ഓർമ്മയ്ക്ക് കിടക്കട്ടെ. രജീഷ് എങ്ക കൊടു. He's here. Bonda முடிய கூடாது. There is bonda. I want to eat that bonda and then talk. Anybody else? Va. Muthu Krishnan. Muthu Krishnaraj. Sorry. அவ்வளவுதானா? 1 second. Yeah. निकल रहे हो क्या?
I will go to temple.
Okay. Wonderful.
We will talk
Pleasure.
about
We are connected.
Yes.
We are connected for life. I know that. We are connected for life. Because I know